Hello, my name is Sarah Anjum. I am a junior microbiology major and I'm an undergraduate research scholar here at the Honors College of Texas Tech University. And today I will be presenting my research over the hepatic effects of fish oil and UCP1 inactivation in diet-induced obese female mice. So as most of us know, obesity has become an epidemic, affecting more than 1/3 of American adults. 70% of these patients who struggle with obesity also have non-alcoholic fatty liver disease, which is an accumulation of excess fat and inflammation in the liver. This disease is characterized by a spectrum of hepatic pathology shown here. It involves a progression to simple steatosis, then steatohepatitis. This is a problem because there are many links to non-alcoholic fatty liver disease and mortality. Omega-3 polyunsaturated fatty acids like eicosapentaenoic acid, or EPA, have proven to be anti-inflammatory and are known to lower bioactives and triglycerides. In a previous study done with high-fat fed male mice, EPA targeted UCP1 in brown adipose tissue, in turn reducing obesity, inflammation, and improving hepatic steatosis by non-shivering thermogenesis. The goal of this study was to investigate the role of UCP1 on hepatic steatosis in female mice fed a high-fat diet supplemented with EPA. So in this study, 12 wild-type female mice were fed a high-fat diet, half of which were also given EPA supplementation. The same diet was fed to 12 UCP1 knockout female mice. Their initial body weights were taken, and after 14 weeks, final body weights were taken as well as liver tissues collected and analyzed. Our hypothesis was that based on previous data, we thought that EPA supplementation would be beneficial in targeting pathways of beta oxidation and de novo lipogenesis in UCP1 knockout mice fed a high-fat diet. To see the effect of diet and genotype on the mice, we ran a two-way ANOVA. As you can see in the body weight graph, there was a significant interaction, but no significant difference among the groups. Looking at figure C, which are the images of stained sections of the liver for each group, we can only see lipid accumulation in knockout high fat and none of the other groups, indicating that EPA may have had some effect on the liver. However, the triglyceride quantification shown in figure B only indicated significant increases in the knockout high fat group and no effects with EPA. Next, we looked at the expression of genes related to fat synthesis to better understand the differences in triglycerides. Our results show that EPA significantly reduced FASN with trending decreases in ACACA in the wild-type group. Next, we examined genes related to beta oxidation. Both PPARA and CPT1A were upregulated in wild-type EPA when compared to wild-type high-fat. However, we did not see these effects in the knockout groups. There were no significant differences in CPT2. In conclusion, although we saw no differences among the groups for body weight, we did see significant increases in hepatic steatosis in the UCP1 knockout groups. We also saw that EPA benefits hepatic lipid metabolism in wild-type groups. In future studies, we hope to explore alternative methods for quantifying triglyceride accumulation so that we can determine if EPA is effective at reducing hepatic steatosis in this model. I would like to thank all the members of the NIOR lab for their guidance in this ongoing project, as well as NIH for funding this study. Thank you for listening.